Assalamualaikum and hi everyone. My name is Muhammad Syarul Azmi bin Ma'arifin, S48389. I'm a student of Bachelor of Science Agrotechnology Aquaculture from Faculty of Fisheries and Food Science. My research title is Determining Health Status and Effect of Captivity on Horseshoe Crab, the Tachyplegigas in Balok via Hemolymph analysis. Horseshoe crab is a type of marine arthropod. There are 4 species of horseshoe crab, one in the North America and 3 in the Southeast Asia. In Malaysia, all 3 Asian horseshoe crab are available. Horseshoe crab live on the muddy, flat and sandy bottoms in the shallow coastal water. They have not changed much for the past 455 million years, hence the nickname living fossil. Habitat degradation due to anthropogenic activities led to depletion of horseshoe crab population around the world. Horseshoe crab in Malaysia are facing unmonitored harvesting, exposure to pollution and habitat destruction. Problem statement for the research is several anthropogenic activities such as fish jetty construction. Recent study by Nelson 2016 also show increased metal concentration of cadmium in the sediment and sulfide in the water were observed. Captive breeding and rearing is considered as the best approach for restocking horseshoe crab population. Prolonged captivity might affect the health of horseshoe crab. Horseshoe crab plays a vital role in human medicine, especially production of vaccine and injectable medication for past 40 years. Detection and essential safety tests for the safe manufacture of all injectable medication. Study shown by Levin et al. 1968 show a new technique for endotoxin detection had been developed using the blood of the horseshoe crab. Pantai Balok are one of 2 places known for spawning activity of the Malaysian horseshoe crab. Heavy metal contamination of cadmium and selenium in the sediment and hydrogen sulfide in the water increased considerably. This change reduced the Tachyplegigas spawning. Objective of this research is 1. To determine hemolymph parameters in wild Tachyplegigas collected from Balok. 2. To compare wild Tachyplegigas data with retrospective data from captive Tachyplegigas previously collected from Balok. The method I used for the experiment is horseshoe crab were collected from Pantai Balok and morphometric parameters were recorded. Then, hemolymph were collected from the joint between bent breastplate and plastron using a plastic syringe with 22G needle. Extracted hemolymph were diluted in pre-chilled marine anticoagulant, and then amebocyte density, viability, and morphology were estimated using hemocytometer. Method for analysis. Hemolymph data used in this study were amebocyte density, viability, and morphology. For morphology, we have 3 shapes, which are contracted, granular flattened, and degranulate flattened. 2 groups were compared, namely Group A, which is primary hemolymph data collected from wild Tachyplegigas, n equal to 3, which is we use 3 samples, and Group B, a secondary retrospective data from captive Tachyplegigas. We use 10 samples, had in captivity for 6 months. Amebocyte density was measured in cell 10 to the power of 7, while viability and morphology were measured in percentage. For the statistical analysis, normality tests were carried out to check if data were normally distributed. Multivariate analysis of variance, MANOVA, was carried out to compare Group A and B in the terms of amebocyte density, viability, and morphology. For the result, normality tests show that all the data were normally distributed, except density data of captivity group. MANOVA test was then carried out to compare the 2 groups. From the analysis, mean cell density of wild and captive group were 2.1 17 to the 10 to the power of 7 and 0.9 times 10 to the power of 7 respectively. Mean cell viability of Y and captive group were 100% and 57%. Percentage of contracted cell morphology in Y and captive group were 27% and 56% respectively. Percentage of granular flattened cells in Y and captive group were 57% and 2%. Percentage of degranulated flattened cells in Y and captive group were 60% and 43% respectively. Analysis showed that Y Tachyplegigas has significantly higher amoebocyte density and viability than captive group. In terms of morphology analysis, Y group shows significantly lower contracted and degranulated flattened cells, while it shows significantly higher granular flattened cells. Figure 1. Amoebocyte cell density of Y and captive Tachyplegigas data is presented as mean of replicate with error bars. Figure 2. Amoebocyte cell viability of Y and captive Tachyplegigas data is represented as mean of replicate with error bars. Figure 3 shows that amoebocyte cell morphology of Y and captive Tachyplegigas data is presented as mean of replicate with error bar. As the conclusion, the high percentage of granulated flattened cells in Y group might indicate that they were exposed to environmental stressor. Captive group might be immunocompromised due to high percentage of degranulated flattened cells. Y group shows significantly better health status than captive group, which show the negative impact of captivity. For future direction and recommendation, due to the fact that horseshoe crab is an endangered species, research was done on a very small sample size. However, high standard deviation in morphology data of white Tachyplegigas samples collected affected the result. Hence, the larger sample is needed in future and animals shall be immediately released afterwards. This is my Gantt chart. My project activity, I start sampling in March and running the project in March and April, and after that I do a data analysis. This is the reference I use for my project. I think that's all from me. Thank you for watching my videos.